Hello everyone, welcome to Enthralling and Fascinating Facts. Today we will learn more about some enthralling and fascinating facts about cells. So stay tuned, watch the video till the end, and if you like the video and find this useful, please do like, share and subscribe. Thank you. Hello everyone, today we will start off with a new organelle which is known as the Golgi apparatus. It is one of the most important organelle in a cell, and also it is included in the endomembrane system. Now if you want to know more about endomembrane system, you can click on the link which is provided in the description box, and from there you can fetch a lot of interesting information about the endomembrane system. Now moving on, Golgi apparatus or Golgi complexes was first observed by Camillo Golgi in 1898 in nerve cells of a barn owl. Occurrence, it occurs in two forms. One is the localized form, another is a diffused form. In localized form, it occurs near the nucleus of the vertebrate cell and mostly all the animal cells. In diffused form, it is scattered in the cytoplasm and not easily distinguishable. The structure of the Golgi apparatus. It is in form of series of flattened membranous sacs called cisternae, tiny vacuoles called vesicles and large vacuoles. Cisternae, these are flattened sac-like structures stacked on one another and arranged in parallel rows. Vesicles, these are small droplet-like structures, and vacuoles, they are large, spacious rounded sacs present on the maturing phase of the Golgi apparatus. Now we will talk about the most important part of the Golgi body, that is the forming phase, which is also known as the cis phase, and the maturing phase, which is known as the trans phase of the cisternae. The Golgi apparatus appeared to have a definite polarity. The two ends or faces of the cisternae called the forming phase or cis phase and the maturing phase trans phase are quite different from each other. How they are different? Well, the sacs on the forming phase differs from the sac on the maturing phase in thickness, contents and a degree of vesicle formation. It appears that the materials are transported from the cis to the trans. The forming on the cis phase is received end of the Golgi apparatus. It is usually located near the endoplasmic reticulum. If you want to know more about endoplasmic reticulum, I have provided a link in the description box. From there you can know more about the endoplasmic reticulum. Materials move to the cis phase in transport vesicles that bud off from the ER. These vesicles fuse with cis phase and empty their contents into the interior lumen of the Golgi apparatus. The ER synthesized molecules then pass through the channels of Golgi apparatus cisternae. The vesicles keep budding off from the cisternae edges to the next sac, so on till they reach the maturing or the trans phase. The maturing or the trans phase is a discharging end of the Golgi apparatus. The cisternae keeps breaking up into secretory vessels and vacuoles at the maturing phase to reach other locations of the cell, or they fuse with the cell membrane, releasing their contents outside the cell. Now the functions of the Golgi apparatus. The most important function of Golgi apparatus are collection, packaging, secretion and transport of molecules synthesized at one place in a cell to the other location in a cell. The proteins and lipids manufactured on the rough and smooth ER membranes are released into the internal compartment of ER. From here, these molecules are transported by vesicles to the cis cisternae. The vesicles fuse with the cis cisternae. As they pass through the lumen of cisternae, they are modified and packaged into secretory vesicles. The common modification is the addition of short sugar chains forming glycoproteins when sugars are added to proteins and glycolipids when sugars are bound to lipids. Many a times, the enzyme present in Golgi apparatus modify the existing glycoproteins and glycolipids made by the ER by cleaving or adding one or more sugars. Depending on the target site, specific groups are added. The modified or altered glycoproteins or glycolipids collect at the maturing phase or trans phase of the discharging end of the Golgi apparatus. Here, at the trans phase, the cistern pinch off small vesicles containing glycoproteins and glycolipids which move to their proper location. They may move to the other location of the cell or fuse with the cell membrane, releasing the content of the internal environment. The Golgi apparatus is closely associated with the ER in structure as well as in functional aspects. Now the Golgi bodies transport and chemically modify materials present in the cells like protein, lipids and sterols. It stores, condenses and packages the material to form in secretory vessels. The vesicles containing secretory molecules get pinched off from the channel of Golgi body and then move to the surface of the cell and discharge the contents to the exterior. Number 3. To add the carbohydrate component to the protein received formation of glycoprotein, the carbohydrate chain acts like a maker on the surface to direct them to the correct destination. To carry out membrane transformations, the membrane of secretory vesicles are similar to plasma membrane. These are constantly incorporated into membrane when they discharge the contents, adding new stretches to the membrane. To form the cell wall in plants, Golgi complexes form all molecules required to form cell plate at the center of the meiotic spindle during mitosis. The formation of lysosome, a very important function of the Golgi complex. Now with this we end the discussion about the Golgi complex. If you have any doubt, feel free to write them in the comment box. I will surely try to clear all your doubts. Thank you everyone for watching this video till the end. If you find this video useful, please do like, share and subscribe and hit the bell icon if you want to receive the notification of my